So this is Unit 11, DNA, the Blueprint of Life. DNA stands for deoxyribonucleic acid, DNA. We're going to talk a little bit about the history of the discovery of DNA. So Irwin Chargaff makes a discovery about the base pairing rules of DNA known as Chargaff's rule. The amount of guanine is equal to the percentage of the amount of cytosine, and the amount of thymine equals the amount of adenine. So this is true for every DNA strand, and because of that, he realized that A and T bond together and G and C bond together because he found those percentages to be the same in every DNA. Around the 1950s, 4 scientists were in a competition to build a model of the structure of DNA, and it's kind of like a race to the moon, but for DNA. Rosalind Franklin and Maurice Wilkins, those were colleagues at King's College in England. And James Watson and Francis Crick, those were colleagues at Cambridge University. In England, Rosalind gets the first X-ray photo of DNA, and there are 2 forms, A and B, and it shows the helical pattern. It also shows that they could measure, when you look at it, you could kind of measure these difference in here, and you can make measurements here. So it also kind of looks twisted, and because of this was the best X-ray photos of the time of DNA, and because of this, you could really kind of make an accurate model. At another university, James Watson is working with Francis Crick on another project. They were model builders. Watson is obsessed with DNA and starts to research. James Watson gets a hold of Rosalind's DNA discovery. So he gets a hold of her picture, and because they were model builders, they were able to successfully build the first model of DNA and get credited with it in 1953. 1962, James Watson and Francis Crick and Maurice Wilkins wins a Nobel Prize for their work with DNA. Rosalind Franklin cannot get a Nobel Prize posthumously. She died due to her life's work exposing her to increased levels of X-rays. Because she worked so much with developing X-ray, we know that X-rays can cause alterations in your DNA, can cause cancers, and she was exposing herself so much she died at a very young age, in her 30s. We're going to talk a little bit about the structure of DNA. So DNA codes for your genes or traits. It's made of repeating subunits called nucleotides. So what is a nucleotide? It's made up of 1 phosphate, 1 deoxyribose or sugar, and a base, nitrogenous base. So here's the phosphate, here's the sugar, and then here's the nitrogenous base. The nitrogenous base is, there's 4 different categories, which is adenine, thymine, guanine, or cytosine. Those are the 4 different choices. So purines versus pyrimidines, the 2 categories of bases, one it has 2 rings and one is just 1 ring. So purines are the 2 ring forms, which is adenine and guanine. So here are the 2 ring forms, 2 rings. You can see the 2 rings. And they will bond to a 1 ring pair. So these are just 1 rings. So thymine and adenine bond together. They fit kind of like a puzzle piece, and cytosine and guanine bond together. So the pyrimidines are 1 ring, cytosine and thymine. Purines are 2 rings, adenine and guanine. So for Chargaff's rule, Chargaff kind of shows that adenine and thymine will go together because they're found in the same percentage. So, let's just say this is like, go back here. So let's just say if this was, 15%, they also found this to be around 15%. And so because of that, he could deduce that guanine and cytosine were in the same percentages, therefore they bond together. Same thing goes with adenine and thymine. The sides of the DNA ladder are the phosphate and sugar, that we call that the backbone. And they're held together by hydrogen bonds. So you can see these hydrogen bonds. These are the hydrogen bonds. So if we think of the base pair rule here, one side we have A, T, A, and so the complementary one for A would be a T, the other side would be a T, an A, a T, an A, a G, T, A, C, G, C, C, C. Think of the bases of DNA like letters. Letters form words, words form sentences, and we have endless combinations of DNA. Last thing here, there's three key features of DNA. It's complementary, which means the nitrogenous base pairings complement one another. So the A's and the T's will fit together, and then the C's and G's fit together. That's complementary. It also runs what's called anti-parallel or in opposite directions. So if this goes from 3 to 5, this one goes from 3 to 5. You can see the opposite directions there. And it is double helix or 2 strands. So last thing here, let's see if you can, what you know about the DNA. DNA stands for, what is the shape of DNA? Who established the structure of DNA? Adenine always pairs with what? The sides of DNA ladder are deoxyribose and what? Guanine always pairs with what? What is the complementary sequence of the one that's given? The two sides of DNA are held together by what kind of bonds? DNA is composed of repeating subunits called what? And what are the 4 base pairs that make up the rungs of the DNA ladder? I'd like you to find the answers of these and put in your notebook. And that's it.